Hello and welcome to today's webinar, focusing on the latest benchtop sequencer from Illumina, the MiSeq i100 series. My name is Helen Bristow. I'm editor of The Pathologist, and I'm delighted to introduce our expert speakers for this topic, who are Jennifer Stobbs and Valentine Alcanis. Jennifer is Senior Product Marketing Manager at Illumina, and Valentine is Staff Segment Marketing Manager at Illumina. And with that, let's get the presentation started. So presenters, over to you.
Good afternoon, everyone. I'm Jen Stobbs. I'm a Product Market Manager at Illumina, and I've got the pleasure to introduce one of our newest product launches today, the MiSeq i100 series. And so today we'll be talking to you about the new instrument, but also some of our oncology workflows that are enabled on this new instrument. So recently we have launched two new next generation sequencing instruments. They look the same from the outside, but they work slightly different on the inside. But these are our simplest and fastest benchtop sequencers that we have available. Utilising some of our new XLEAP SBS sequencing chemistry, with some fantastic improvements in the way the instruments work and the benefits to your lab. So this really will empower every lab everywhere to be able to take on next generation sequencing and discover more. So what have we launched? So with the two new instruments that we've announced is the MiSeq i100 and the MiSeq i100 Plus. They do look the same, but inside the MiSeq i100 system can do 5 million reads or 25 million reads, and the MiSeq i100 Plus can now do 5 million, 25 million, 50 million or 100 million reads. So this really does expand the output of the benchtop sequencing instruments, meaning that you can do small targeted panels with a really fast turnaround when you've got low sample numbers by utilising the 5 million read cartridge. Or if you want to batch up your samples to make it the most cost effective way, you can go right up to 100 million reads. It gives a great amount of flexibility to be able to do lots of different sequencing experiments. So currently we've announced we will be shipping in December the MiSeq i100 Plus as an early access, and then it will be fully available as a full release from March 2025. Available at launch, we will have the 5 million and the 25 million read flow cells. And then in mid-2025, we will have the 50 million read and the 100 million read flow cell available as well. And then we will launch the lower throughput instrument, the MiSeq i100. So two different stages, but by mid-year, you should be able to have the full capacity of everything we have available. So what does that actually mean in terms of samples and what you actually want to run on a sequencing instrument? So today we're mostly talking around targeted gene sequencing. So on that smallest 5 million read cartridge, that if you wanted to do a targeted gene panel, it needs around 4 million read pairs per sample. You'd be able to do 2 samples, but that might be advantageous if you've got an urgent turnaround. Right through to the other end of the spectrum is the 100 million read cartridge. So with the same size panel, you could then do 50 samples, which gives you a lot of flexibility depending on how many samples you've got. So the MiSeq i100 series, what makes it so different from other next generation sequencing instruments is that it is incredibly simple to use. We've really streamlined the way the system operates with various different features, including not having to thaw any of the reagents. It doesn't need to be stored in fridges and freezers. We've validated, as research products, we've validated 18 sample to analysis workflows. So it's all simplified, it's just push button. And it's much, much faster. Next generation sequencing with older SBS chemistry depending on the flow cell that you chose to use, could take a long time, 2 to 3 days. The new sequencing chemistry is considerably faster, up to 4 times faster, which means you can get your sequencing information very, very quickly. But having a simplified workflow and a much faster workflow, it still needs to be high quality. Now, the reason MiSeq has been so popular over the years is because it does produce reliable, high quality results. So the new systems had to be the same, if not better, in terms of data quality, because that's obviously crucial. So the purpose of the introduction slides is to go through, sort of demonstrate these advantages before we move into workflows. So the MiSeq i100 series of instruments means that you can expand your applications and scale more effectively. So you can still do things like targeted gene panels, which are obviously quite popular in the lower throughput flow cells. But you might want to move towards larger targeted gene sequencing as well, or you might want to start thinking about things like RNA-seq to see what the impact of those DNA changes are on the transcriptome. And this is enabled now by having the MiSeq i100 Plus that has the capability to go up to 100 million reads. It means that you've got one instrument, but you can do a much bigger breadth of different types of experiments with your samples. So first we're going to sort of dip our toes into the simplicity of the instrument. And it isn't just the sequencing instrument, we've simplified the workflow right from the moment you actually receive the instrument, right through to the end of data generation. So the whole process has been simplified. So just as a summary, we've streamlined the setup. So actually being able to do the library preps and add those into the flow cells is so much easier to do now. It's simply 3 steps, which can be done in under 20 minutes. It's very, very user-friendly, and there's no off-instrument things to do. As soon as your libraries are prepared, add it to the flow cell cartridge, and then you've got 3 steps. There's minimal hands-on time, so a lot of the sequence and chemistry reactions happen on the instrument. So denaturation of the libraries happens on the flow cell, and also cluster generation. So you don't need to have any additional cluster generation instruments with Illumina systems. It all happens on board. We've also integrated data analysis into the sequencing instrument. So we have a Dragon secondary analysis platform. So Dragon, basically it takes the sequencing reads that are generated by the sequencing instrument and aligns those to a reference genome. And Dragon is, well, it's actually a hardware tool, but it also does the software analysis as well. But it's so much faster because of the way the hardware works. So that's all integrated into the sequencer now. But you do also have options to use things that are in the cloud to be able to get additional bioinformatics pipelines. But that does mean that it's much easier to use. You don't need any bioinformatics expertise to be able to get on board with next generation sequencing. Maintenance is also simplified. So it's not just the sequencing runs, it's everything that happens around it. So there is a lot less maintenance to do now. And that's because a lot of the fluidics are contained within the flow cell. So you don't need to do washes in between runs because everything happens within the flow cell and then you dispose of the flow cell. So there's no internal tubing to be able to have to prevent contamination between runs. So those wash steps are all removed. And then when we talk about disposal as well, so once you've finished with the flow cell and the cartridge, you can actually take the plastic pieces apart and recycle the plastics just in standard plastic recycling. And a key one as well is that a lot of sequencing chemistries previously have contained formamide, which is a fairly nasty chemical to have to handle. So we've removed that from the new sequencing chemistry. So you don't need to worry about how to handle those now. We've also simplified storage. So the core consumables, the flow cell and the wet cartridge, They are now stored at room temperature. They are transported from our warehouses at room temperature, and they can be stored simply on the shelves. So you don't need to worry about the costs and having to make sure that you've got enough fridge and freezer storage. It's just all stored at room temperature now. And that simplifies things in terms of run setup as well. So now if you receive some samples on the morning and you want to get the sequencing on that afternoon, you don't need to wait for reagents to thaw and worry about having to mix them, because that happens on the instrument. It also avoids running consistencies because you don't need to wait for things to thaw. And if you're not sure whether they have thawed or not, it's no longer a problem. That also means that you can run samples faster. So you don't have to plan ahead for the following day to make sure everything is available and thawed, available at the right temperature. As soon as you have your samples ready, you can go. The shelf life is guaranteed for 3 months at launch. We are looking to expand that. Obviously, we need some time to pass to be able to fully test and make sure that it is robust beyond 3 months, but we are planning to go beyond that if possible. This is what the flow cell and the cartridge looks like. So on the left-hand side, this is a 25 million read flow cell. So inside of all of the white plastic is where all of the fluidics are contained. And the gold chip in the middle is actually a CMOS chip. That's where the magic happens. So there are two rectangles there that you can see. So in the larger flow cells, you would have two CMOS chips, which is the difference between the lower throughput instrument and the i100 Plus. The i100 Plus can read both of those positions, whereas the i100 only reads one. But the CMOS chip is what does it generates the sequencing reads. So at the bottom of the flow cell, you can also see there's 4 different wells. So the left-hand side one just says insert library here. So as soon as your library is prepped, you pipette it into, you make a dilution and you pipette it into that well and you slide it into the instrument. It's incredibly simple. The right-hand cartridge is the wet cartridge, so it contains all of the sequencing reagents. So what happens is the instrument takes the wet reagents and brings it into the flow cell cartridge, because the flow cell cartridge contains a lot of the lyophilized enzymes. So the wet cartridge is used to rehydrate all those lyophilized enzymes to make the sequencing reactions happen. Next, I'll touch on some of the speed improvements from the workflow. So this just kind of compares how those advancements compare to our previous next generation sequencing, the old MiSeq instrument that we have. So at the top there, you can see the MiSeq i100 series, the number of touch points, the number of times you have to interact with the instrument has been dramatically reduced. And the workflow time as well. So actually setting up the instrument is more efficient. The sequence and run time is considerably faster. And then analysis time as well, because there are drag and analysis tools on board, but also in the cloud. That is much faster as well. And when we compare that to the MiSeq V2 chemistry or the MiSeq V3 chemistry, the number of touch points there are much higher. And that is important because it means that potentially the more touch points there are, the more chances there are for the user to make an error. So we've tried to streamline that as much as possible, so it's more reliable. But also when you look at the sequence and run times, so on the V3 chemistry, which runs a 300 cycle run, then it takes a lot longer. So we've speeded everything up to make sure you can turn around faster. Dragon secondary analysis really speeds things up as well. So compared to BWA, G80K pipelines, which are some of the things that people develop in-house, Dragon can do a lot of those workflows in a much faster way, and it's incredibly easy to use. So we've got some onboard analysis options that are available, and we've got some in the cloud. But what's important is you don't need bioinformatic expertise or command line training to be able to pull those off. It is push button. It's incredibly efficient. So actually you can get some of the results in as little as 2 hours. So because of the way the sequencing now happens, you can actually, within 2 hours, you can find out how balanced the libraries are. And that is important for people who are doing QC runs for larger instruments. There's a 4 times reduction in those manual touch points, which obviously prevents errors, and then faster data transfers from compression. So with the Dragon analysis tool, it does also incorporate data compression, which means that the final variant reports that you get can actually be stored in a much smaller space. So you don't need as much server space to store the genomic information that you're generating. Obviously, it's great to have a simplified workflow and that it runs much faster, but what about the data quality? So with Illumina, we have launched 3 new instruments now that incorporate the XLEAP SBS sequencing chemistry. So this is a much improved version of the old SBS that we used to have. So we launched the NovaSeq X that features a version of XLEAP chemistry. And then we brought the XLEAP chemistry onto the NextSeq 1K and 2K systems. And now we've advanced that SBS chemistry even further onto the MiSeq i100 series. So the chemistry is slightly different between the 3, but it is huge advantages over our previous chemistries as well. And the reason for the 3 different instruments is the amount of output that you want to generate. So the MiSeq i100 series is great for things like targeted gene panels. If the NextSeq range is more suitable for things like if you want to start looking at exons or single cell sequencing. And the NovaSeq X series is more suited for things like higher throughput whole genome sequencing, or if you're doing large projects that are more in the proteomics space. So we now have the 3 instruments that are suitable across multiple different types of research experiments. But this is all based on Illumina SBS sequencing chemistry. And in the MiSeq space, it's been a renowned instrument for a very long time. We have over 10,000 MiSeqs installed across the world. It's an incredibly trusted instrument. It's been cited in over 160,000 publications, which shows the number of people that have relied on MiSeq sequencing. It certainly is still a favourite out there. It is still a very popular instrument. But that's because people have trusted the MiSeq and Illumina sequencing for a long time. So we need to make sure that we continue that with the MiSeq i100 series. So the quality improvements, along with the speed and efficiency, all comes from these XLEAP SBS chemistry advancements. So it really does achieve a higher level of accuracy and performance on the new series. So it is a novel polymerase that does the sequencing chemistry, and it is different to what is available on the NextSeq and the NovaSeq. But these advantages make the reaction and the enzymes, it's much more stable. So it's 50 times more stable in solution, but also in terms of how we can actually ship that. That's what enables us to be able to ship at room temperature and store at room temperature because of these advantages. So when it comes to quality, so with the XLEAP SBS chemistry that we have on the MiSeq i100 series, on the left-hand side, you can see a comparison of the mean error rate when we compare in grey the MiSeq v3 chemistry, which was the previous instrument, and then in orange the MiSeq i100, which is the new sequencing chemistry. And the error rate is dramatically reduced there, particularly on read 2. And on the right-hand side, you can see the average, the mean Q30 score has improved between the MiSeq v3 and the MiSeq i100 series, again, particularly on read 2. So the quality improvements are much, much better. And when it comes to our oncology panels that we have, again, Valentin's going to talk about that in more detail. But this is a comparison between using the Pillar oncoReveal myeloid panel to compare the data that's generated from the old MiSeq and the new MiSeq to make sure that there is a high correlation between the 2 systems. What you can see in the table on the right-hand side, particularly there is an improvement in the Q30 percentage, but particularly the error rate has dramatically dropped. What you can also see is the yield has improved even from the same output flow cell. And this is because the new flow cells are a patterned flow cell, whereas the old MiSeq was random. And this has advantages in terms of being able to generate more data from the same space. So you can actually get more usable data from the sequencing system. So I'm going to stop there and I'm going to pass over to Valentin now.
In the second part of the webinar through how the MiSeq i100 can help the oncology research in every lab everywhere. So the presentation will be split in 4 main parts. The first one is just a general introduction on genomic solutions for oncology research, highlighting the importance of next generation sequencing in that field. The second and the third one will be 2 pieces of the portfolio, the Pillar Biosciences oncoReveal and the Truth Hereditary Cancer, and how they tie up together with the MiSeq i100. And finally, we'll speak about data analysis for oncology research. So let's start with genomic solutions for oncology research. Oncology research spans across different areas of knowledge, from germline, early detection, to therapy selection and monitoring. And at Illumina, we offer solutions that are suited for the different areas of interest, from single gene testing to comprehensive genome profiling. But also, we offer solutions in the new emerging areas like whole genome sequencing. Through the years, and as sequencing cost has been declining and evidence and ease of use has been improving, we have saw the rise of different omics in the oncology research space, now becoming highly popular proteomics, transcriptomics, and whole genome. But in this webinar, we will be highlighting how the MiSeq i100 is the perfect partner for disease-specific hotspot panels. So, why this is important? Here, we're able to see the number of biomarker-derived indications that were approved from 2003 in the United States for non-small cell lung cancer. And there are more than 45 indications that has a biomarker linked to that indication. You are able to see here in this part of the presentation the different biomarkers and plotted with the different indications for non-small cell lung cancer, highlighting the importance of next-generation sequencing and genomics in the oncology research field. But not only in non-small cell lung cancer, there's a growing list of biomarkers that are highly important in other tumor types, like, for example, BRCA1 and BRCA2 genes in breast cancer, or BRAF, ERB2, and KRAS in colorectal cancer. Even though there are also biomarkers that are important independently of the tumor type. We have NTRK, TMB, MSI, BRAF, and RET that are highly important to be studied independently on the solid tumor type. So they are considered tumor-agnostic biomarkers. At Illumina, we do offer a wide portfolio that covers the different needs that oncology research may have around the globe. From targeted panels to whole genome sequencing, we get you covered. And the portfolio spans out in 3 main parts: solid and soft tumors, hematological tumors, and liquid biopsy panels. Here, the MiSeq i100 is the perfect partner for targeted panels, both in the somatic and in the germline testing area, for also both solid and soft tumors and hematological tumors. But the Illumina portfolio does not only consist in a combination of systems and applications, it also covers the whole workflow and the whole continuum of the samples analysis. From library prep, sequencing, secondary analysis, and tertiary analysis, we offer a connected ecosystem that can work with the complete workflows in the oncology research space. We provide it as a single provider in a connected ecosystem with the demonstrated quality that has been highlighted in the previous part, and also offering the best-in-class support from our support team. So let's take a look on the Pillar Biosciences portfolio. We have came up with an agreement with Pillar Biosciences to boost up our oncology portfolio, providing new and up-to-date content for the oncology research space, providing basically amplicon-based assays for oncology. We offer it in a seamless customer experience where you can rely on Illumina to take care of all of the needs, from sales to support. But also, thanks to that partnership, we're able to offer oncology content that meets the needs of the researchers worldwide, from small panels to comprehensive genomic profiling. We are able to cover the complete spectrum of the different sizes of applications that one can choose for oncology research. So this partnership has bring it up into a point that we can offer fast, simple, and cost-effective next-generation sequencing panels worldwide. We are able to offer simple library prep workflows that consist in a single tube, easy and fast library prep applications that offers also sensitive and robust chemistry, where we can achieve down to 1% variant allele frequency without the use of UMIs, and even with limited DNA input or poor sample quality. And also we are able to reduce the cost of labs thanks to the reducing of the no-calls. And when we pair this portfolio with the MiSeq i100, we are able to offer single-day sample to result, thanks to the unprecedented speed of the MiSeq i100 sequencing system. So how does a workflow look like? First, we start with the library prep part. We need to take the DNA and RNA and convert it to libraries. And in this part of the workflow, we can work it both manual or in automated fancy. We can work with third-party library prep liquid handle providers like Beckman Ingenious to perform the libraries in an automated way. In less than 7 hours, we are able to construct the libraries with the Pillar Biosciences portfolio. Then, as my colleague Jen has explained, we are able to make a sequencing run with 3 steps only and in less than 20 minutes. And with a total runtime of 7 or 8 hours, we will be able to get the sequencing results that then can be streamlined directly to drug and secondary analysis to have the final results from the secondary analysis in less than 2 hours. So what does it mean? It means that now we're able to provide complete workflows for oncology research in less than 17 hours of total assay time. So we are able to provide results in a single day. So we have already taken a look previously on how the sequencing works and how the sequencing setup is performed. But here we want to highlight also how the library prep part works from Pillar Biosciences. And we are comparing a conventional amplification workflow with almost 10.5 hours and multi-day workflows that contains several steps with the easy and simple workflows from Pillar Biosciences that can be completed in a single tube with just 4 main steps in less than 6.5 hours. And which is the portfolio that we have in collaboration with Pillar Biosciences? Currently, we have validated 4 main panels in the MiSeq i100 system. We have validated the multi-cancer panel that suits for lung samples, melanoma samples, thyroid samples, colorectal, or gastrointestinal stromal tumor samples. We have validated the BRCA1 and BRCA2 panel, the MPN panel, and the myeloid panel. All of them provide great guideline gene coverage based on targeted amplicon assays. And also, all of them share the same characteristics. It's an easy, single tube manual workflow that can be automated on a Beckman Ingenious with less than 24 hours of total turnaround time. And also, regarding input material, they can work with very little DNA and RNA, from 5 to 20 nanograms of DNA and 10 nanograms of RNA. So how does it look like when you mix it up with the MiSeq i100? Here, we want to show that the combination of the MiSeq i100 and Pillar Biosciences portfolio offers faster runtimes and more flexibility than any other benchtop sequencer. For example, we are able to sequence together 32 samples of the multi-cancer panel in the 25 million flow cell that Jennifer has explained in less than 7 hours. But also, we are able to sequence up to 48 myeloid samples in less than 8 hours in the 100 million flow cells, offering huge flexibility for oncology research around the globe. Now, let's switch gears and let's switch also technology and let's speak on how can impact hereditary cancer research. And we'll be showcasing how the TrueSight hereditary cancer research portfolio works and looks like. TrueSight hereditary cancer portfolio, it's living on the Illumina DNA prep. And let me explain how the Illumina DNA prep with enrichment works. It's a fast and robust workflow with just 2 hours of hands-on time, and it can be completed in just 6.5 hours of total assay time. It can take different sample inputs. It can take germline DNA, blood, or saliva. Thanks to its technology, it normalizes the libraries with a bit-based normalize. And speaking about content, it comes with 130 genes and 125 SNPs, 48 of them being used for identification and 77 of them being used for polygenetic risk scores. And also, it comes in an integrated solution, meaning that it can be work with Dragon for secondary analysis and the tertiary analysis options. But how does the technology works? Here we are showing how we can generate libraries in less than 2.5 hours that then we will need to enrichment. But let me explain how the technology works. Basically, you mix up DNA with the enrichment-based link transposons that cuts and indexes the DNA. So at the end of this 2.5 hours process, you're able to have indexed libraries. Those indexed libraries, we need to enrich them. So we need to add the biotinylated probes from the TrueSight hereditary cancer panel, and then we can enrich them using streptavidin, and we are able to get enriched libraries that are ready to be sequenced. This whole workflow, the 2 parts, takes less than 6.5 hours. So again, we are able to offer the same workflow as with the Pillar Biosciences portfolio. We can prepare the libraries both in a manual or in an automated way, here partnering with Hamilton, and then we're able to sequence them in less than 8 hours to finally streamline the data analysis with Dragon and have the results in less, again, than 17 hours. So it's a workflow that is super fast and is suitable to obtain single-day results. Here we're showing an example also on how many samples we can put together in the same flow cells, and just taking 300x of mean coverage as a standard for this panel, we are able to put 16 samples in the 25 million flow cells, and it will be sequenced in less than 7 hours. But also, if we have more samples and we want to put more samples in the same run, we're able to put up to 64 samples in the 100 million flow cells, and it will be sequenced also in less than 8 hours. It's a system combination that is super fast and super flexible. So then, once we have sequenced the data and performed the libraries, we're ready to jump into the data analysis part. This is how a typical workflow looks like. And as I have explained, first, you need to prepare your libraries, converting DNA into a construct that is compatible with the different Illumina sequencers. You're able to sequence them, in this case, in the MiSeq i100. And then we can do the secondary analysis with Dragon. Once we've done the secondary analysis, we'll receive back a variant calling file, a BCF. That BCF, we need to extract the insights from that file to understand the significance of the different regions of the genome that we have sequenced, and also to understand the significance of the different variants that we have found into that samples. For that, we are able to put the BCF into Illumina Connected Insights, and in this platform, we are able to interrogate different biomarkers, different guidelines, different drug labels, and also clinical trials, to, at the end, be able to generate a report that is extractable and that can be using down the pipe. This might sound complicated. Also, the secondary analysis part. But Illumina has developed an efficient no-touch workflow with Illumina Connected Analytics that basically it's able to offer a no-touch experience from the secondary analysis to the tertiary analysis. And how does it work? Basically, we are able to transfer automatically with a no-touch experience data from the sequencer to Illumina Connected Analytics. There, we will do the secondary analysis, and from there, we will transfer the data to the tertiary analysis platform to perform the interpretation and the insights extraction. All of this can be integrated in a no-touch workflow with a user-friendly user interface and in the same cloud. But also, if users want to work locally, we have also on-prem solutions that can work with your own institution internet requirements. Once we have the data into Illumina Connected Insights, what we can do is to extract the significance of the variants and filter down the ones that are important for us and the ones that are not important for the research purpose of the study. So Illumina Connected Insights connect with more than 55 different sources, like databases like CBIC, CKB, OncoKB, and also you can build up your own private knowledge base. And not only databases, it connects you also with algorithms like Primate AI or Splice AI to be able to understand the significance of a variant on a genome. Also, Illumina Connected Insights offers you the capabilities to understand the significance of a variant. And here we are showing a map that basically it's explaining the classification of the variants based on different parameters. This can be automatized or it can be worked also on a manual fencing. And at the end, once we have interrogated the different variants on the different samples, we are able to extract a report that can live in different report templates and in different languages. And users can define the information that is useful for them and that then can be extracted as a PDF or as any of the different formats that we do offer to extract the information from Illumina Connected Insights. So with that, we finish the presentation part. We hope that we have give you a lot of information on how the MiSeq i100 empowers every lab everywhere. And now we are opening to questions from the audience. Many thanks, everyone.
Okay, so now it's over to the audience who are ready to start the Q&A session. So please do continue to submit any questions you have using the question box on your screen. It's a fantastic opportunity to pick the brains of our two experts today. So the first question. Is it possible to run two FCs independently on the new MiSeq i100?
It's not at the moment. So these instruments are designed to just do one run at a time. So there is flexibility in the output. So that means that if you've got a lot of samples that you want to run, you could choose a bigger flow cell. But also the speed improvements do mean that you can potentially do 2 runs in a day if you'd like to. So it should remove the need for needing 2 flow cells. We did recently hold an event where we had one of our early alpha testers share his experience of how they'd used it. And because some of the runs are only 7 hours long, they had set up a sequencing run on the morning, which was done and completed. And then they were ready to set up another sequencing run that could then run overnight. So the speed improvements now do mean that you possibly won't need to run 2 flow cells at the same time, because you can actually just run one after another and still get the sequencing data very quickly.
Fabulous, thank you. And a question about Dragon. Does Dragon provide a report, a third-part analysis, or do we need a dedicated software for the clinical report?
Yep. So Dragon provides a QC report, meaning a quality report, and also provides VCF, which is the variant calling file. But then if you want to use a tertiary analysis software to extract the insights and understand the significance of the variants, you can use Illumina Connected Insights, or also you can use any of the other tertiary analysis options that are on the market. So basically, Dragon, it's the secondary analysis part where you have your variants and your QC done.
Fantastic. And another question about Dragon. I was wondering if Dragon could be executed in isolation without requiring a previous sequencing. Also, if there's any chance to know its internals. Black boxes may not always be the best solution, even in clinical settings, as we have seen with iron torrent solutions.
So with the MiSeq i100, there is the Dragon on board the system. So it will only process data that has been generated on that system. You do have the option to buy additional Dragon servers, which are standalone, and they can take sequencing data from various different sources and run those analysis apps. I do understand the question around whether you can tweak the internal algorithm. So it is, the advantages of having the Dragon on board is it runs off the technology called FPGA chips, which is what makes it super, super fast. But the other benefits of Dragon is that there are significant quality improvements in the variants that it calls. And that algorithm is actually freely available. So if you do want to reach out directly, I can find some of those links and share the information, because then you can actually look at how the algorithm works. And you can still use that information however you like, and that is free to use that. It doesn't benefit from the speed improvements because it isn't paired with an FPGA chip. So yeah, for a clinical setting, black box isn't necessarily what you need, so you can at least look into the background of that.
And also adding on to that, Dragon can be accessible as a standalone tool. You can check it in Base Space or Illumina Connected Analytics, where you can have access to Dragon and put any sequencing run that you have done previously, and you can analyze it.
Thank you for clarifying that. How many samples can you run with the TSO 500 panel with the MiSeq i100 Plus sequencer? Yeah, so currently in the oncology research space, we have only validated the 4 pillar panels that I've shown you, and also the TrueSight hereditary cancer. There are more panels going to be validated down the pipeline. TSO 500 is not currently validated in the MiSeq i100, so we are not able to run it now. If we just go to the numbers, we know that TSO 500 needs 50 million clusters, and the biggest flow cell of the MiSeq i100 has 100 million clusters. So mathematically, we should be able to squeeze 2 samples in one single flow cell, but we have not tested it. But we can stay in contact directly to look into a detail on that. Thank you. Just for clarity, after sequencing the first flow cell, can I remove it and sequence a second flow cell as the first flow cell data analysis continues?
Unfortunately, no. So once the sequencing is completed, if you're using the Dragon server, you need to wait for the Dragon analysis to finish. However, because Dragon is so much faster and this is a lower throughput instrument, the turnaround times on those are incredibly quick anyway. So you are literally talking less than an hour, some minutes in some cases, depending on the size of the panel that you're using to sequence. So actually, it doesn't make a lot of difference, particularly if you're doing a sequencing run overnight, because the analysis is also determined right at the start of the sequencing setup. So sequencing automatically goes onto the analysis without having to have any user interaction. So that does mean if you're running it overnight, it will have done and finished the Dragon analysis when you're ready to come in the next day. So because it is so fast, it shouldn't actually make a lot of difference.
Okay, thank you. The next question is, how many libraries prepared with TrueSight hereditary cancer can be updated on the MiSeq i100?
Yeah, so it depends on the coverage. If we take 300x as mean coverage as an example, we're able to put 3 samples on the 5 million flow cell, 16 samples in the 25 million flow cell, 32 samples in the 50 million, and 64 samples in the 100 million. It's just the example that I gave in the slides. There are many other multiple combinations in case that you want to go 200x mean coverage, or you want to go even lower at 100x mean coverage. So please reach out directly and we can give you indications on how many samples you can put together based on your concrete example of desired mean coverage of that panel. Thanks.
Sure. And are the different flow cell prices available?
Yeah, so in this case, it's probably best to speak to the salesperson. So if you do reach out, we can connect you on that. Obviously, there's a lot of different flow cells available depending on the type of experiment that you want to run. But on average, I think it's up to 40% cheaper than it previously was with our old MiSeq chemistry. So there are a lot of cost savings to be made there. But obviously, it depends on the flow cells that you buy as to how much it is reduced. So please do reach out and we'll connect you with the salesperson and they can share that pricing information with you.
Sounds good. Next question, do we need a license to use insights? And if so, how should one go about it? Can we have credits for each run?
Yeah. So there's a license for Illumina Connected Insights, but the license has no cost. But there's a cost on the implementation where we need to set up the lab and set every bioinformatics step properly. And it works on accredited systems. So we are able to offer different genome equivalents that contains different samples for the different panels. So depending on the size of the panel, there's a price per sample that we need to take a look at. But please reach out directly and we should be able to connect with a sales colleague that should give you all the details.
Fantastic, thank you. Are there any plans to validate further pathology workflows from Pillar and other companies such as Archer, Sophia Genetics, adjuvant panels?
Yeah, thanks. So we are validating further panels from Pillar Biosciences and also further Illumina panels, but also Illumina is an open platform. So we can sequence any library prep from the different providers. Just connect with the salespeople and we will let you know if we have already done it anywhere in the world or if you will be the first one to do it.
And what are the key data analysis tools that are integrated with the MiSeq i100? And how do they facilitate the interpretation of complex genomic data?
So with the MiSeq i100 instrument, we've talked about the Dragon. So the Dragon on board does have some selected workflow applications that you can do. At the moment, that includes some of like the QC runs. It includes some of the microbiology applications as well. But you can also access everything that we have lots of Dragon analysis apps in base Sequence Hub as well as lots of third-party analysis apps. So there is a lot there to choose from, so that it depends on the data that you want to look at and what type of experiment you're running. But we do have lots of specialists as well who can help give guidance on which one might be the most appropriate. Obviously, genomic data produces quite a lot of data, so it's important to choose the right analysis workflow. But the Dragon on board is a significant advantage, or use Dragon in the cloud for additional apps if you need them.
Great, thank you. And what kind of training and support are provided for labs that are transitioning to the MiSeq i100?
Lots. So we do, Illumina is well known for, we have a huge support network. We have field support, we have online technical support. It's almost round the clock because we're a global company as well. So in terms of training and support, we do have lots of online videos. There is a learn section on the Illumina website as well. But we also host things like training sessions where you can join us in the Illumina Solution Centres to 2 or 3 day training sessions that walk you through everything from the library prep, the sequencing, right through to data analysis. We also have virtual versions of those as well that people can sign up for to learn more. And then we do have regional support teams as well. So if you'd like to have your local field scientists come and visit you in the lab to help you do that, you can do so. But there's a really good starting point. There's a lot of videos that you can access and see how simple it is to get going. But it's also worth reaching to your local salesperson, because they can help you choose the right one, or which one's the most appropriate for your lab, and guide you in the right direction as well. Oh, just to add to that as well, we do also offer a service called Workflow and Design and Evaluation. So for anyone who would like to see what the data looks like from the new sequencing runs, what we can do if you work with the salesperson is to send some of your samples into one of our solution centres where they already have the instruments, and they can do those sequencing runs and send the data back. So if you want to compare a library that you've run previously on a different instrument, or if you haven't done NGS before and you want to see what that looks like, we can do the workflow design and evaluation to generate some initial data on your samples as a proof of concept kind of thing. And the way that usually works is then if you do purchase an instrument following that, we can then discount the cost of it off the instrument. So it's kind of like a try before you buy type thing. So it's worth considering.
Fantastic. That's I'm not seeing any more questions coming through. So do you have any sort of closing message or words of advice for labs thinking of switching to this sort of workflow?
We do have lots of resources, so please do visit Illumina.com. You'll see the banner right at the top that leads you to the MiSeq i100 page. There's lots of resources on there. If you're not familiar with NGS, there is a learn section on Illumina.com where you can find out more about NGS, what the advantages are of moving from other traditional methods like qPCR or Sanger or MLPA. These are all approaches that you can now group together and do NGS instead and get that same type of information from those traditional approaches. So the website is a really great hub, but do reach out to us as well, because the sales team can also bring in the support colleagues to help you work through what is the right option for you or for the experiments that you want to do and the research that you're looking at.
And audience will have noticed that Jennifer and Valentine have kindly added their contact details in the audience chat. So please do make a note of those. And if you think of any further questions after the presentation, please do feel free to reach out. So Jennifer, Valentine, thank you very much for answering those questions and for presenting that information today. If the audience wish to revisit the topics covered today, then the webinar will be available on demand on our website in the next few days. So thank you again for sharing your expertise on the MiSeq i100 series. Thank you to everyone attending, and I look forward to seeing you at future webinars.